Okay, welcome everybody. This is chapter 12 dealing with the nervous system. And as you probably noticed in your textbook, this is probably the thickest chapter you're going to run into. And the reason is that the nervous system does more than any other system in the body. So usually double the amount of material is dedicated to the nervous system as any other body system. Now there's a reason for this. Okay, the nervous system has a lot to do. It has to collect information in the form of stimuli, which are physical changes that happen both inside and on the surface of the body, okay, as well as coming in from places like the special senses, the eyes, the ears, the nose, our balance apparatus, and so on, okay. It has to turn that information into electrical information, and it has to pipe it into the central nervous system, where it makes its way up to the brainstem and then the brain. And when it gets there, it has to be assigned a meaning. Then it has to be combined with all the other information that's coming up to the brain at the same time. And then that information, once it's been categorized, has to be sent in one block to a region of the brain called the frontal lobe, where we decide what we want to do about what's happening to us. And then we generate a command in the frontal lobe of the brain that then goes down through a structure called the brainstem and then to the spinal cord, and then out what are called peripheral nerves that target glands and muscles and other tissues and generate the response to the stimulus. Okay? This is going on all the time. Now, most of the stimuli that we experience don't make it to our consciousness, but they're still dealt with because there are regions of the brain that work with information that we don't need to be consciously aware of. And those regions are the hypothalamus and the brainstem. The hypothalamus lies below a structure in the brain called the thalamus, which we'll get to in a bit, and the brainstem lies below the diencephalon, which is made up of the thalamus, the hypothalamus, the epithalamus, and the subthalamus. Okay? Now, the spinal cord is below that and is a pipeline that brings information up to or down from the brain and the brain stem. Okay? So this is one of the body's two communication networks. This is how the body is able to send information, interpret it, and send out commands that respond to that information in order to enhance our survival and maintain homeostasis. Okay? The two divisions of the nervous system are the central nervous system, which is the brain, brainstem, and spinal cord, and the peripheral nervous system, which is everything else, which would be basically the peripheral nerves and the ganglia, which are groups of nerve cell bodies that lie outside the central nervous system. Okay. Now, the brain, brainstem, and spinal cord are well protected. The brain and brainstem sit inside the cranium, and the spinal cord sits inside the vertebral column. And because they're so well protected by not only bone, but a fluid called cerebrospinal fluid and membranes called meninges, the central nervous system tissue has largely lost the ability to repair itself when it's damaged. And that's one of the reasons why if you have a break in the spinal cord, the information can't go down past or up through the break and make it to and from the brain, okay? And it's why if you have a traumatic brain injury, you lose that brain tissue permanently, okay? Peripheral nervous system is a little different. Peripheral nerves can repair to a small extent, provided certain conditions are met, okay? But it does not have the ability to reproduce. No nerve cell does, okay? With very few exceptions. Functional organization. The sensory nervous system is what takes the stimuli and turns it into electrical information and sends it to the central nervous system. The somatosensory system detects stimuli on the surface of the body and from our muscles and our joints, while the visceral nervous system takes information from our internal organs. Now, of this information, a very small percentage of it makes it to our conscious awareness. Most of it is filtered out by a structure called the thalamus, and that information, even though it doesn't make it to our conscious awareness, if it's critical, it's dealt with by the hypothalamus and the brainstem, which acts as sort of a second brain that controls our subconscious activity. The efferent divisions, or the outgoing divisions, include the somatic motor division, which sends commands to skeletal muscle, and we consciously control that, and the autonomic motor system, which is broken into the sympathetic and parasympathetic divisions, and they target everything but skeletal muscle and are important in maintaining homeostasis. So you can see some of the different divisions here, how they're broken down into both functional and anatomical divisions. Okay? So functional division, what it does, anatomical division, where it's located. All right? A nerve is a peripheral nervous system structure, which is basically a bundle of axons. Now, an axon is simply an outgoing pipe for a neuron that carries the electrical signal to the next cell in a nerve pathway, which could be another nerve cell, or could be a muscle cell, or could be a gland. Okay, the axons may be myelinated, they may not be myelinated, they may be slightly myelinated. It all depends on what nerve we're talking about. And the direction that they carry the signal will depend on the nerve that we're talking about. Some nerves will carry signals only outgoing, which means they are efferent nerves and they only carry commands out to a target. Some nerves will only carry information into the central nervous system, so they are afferent nerves. All right. And then some nerves carry information in both directions, and those are called mixed nerves. Connective tissue that accompanies the nerve includes the epineurium, which surrounds the entire nerve, the perineurium that surrounds fascicles of nerve fibers, and the endoneurium, which surrounds individual nerve fibers. In addition to stabilizing the position of the nerve, it also insulates the electrical information so that there is no short-circuiting of the electrical information in any of the different classifications of nerves, efferent, afferent, or mixed. Okay? Nerves are vascularized. There's a blood supply that runs with the nerve. Nerve tissue requires a constant supply of oxygen and glucose in order to function. Glucose is the only fuel that neurons are able to use in order to make energy. Okay? You can see the structure of a nerve and a ganglion here. Okay? Ganglions are simply clusters of nerve cell bodies that lie in the peripheral nervous system, and of course the nerve, as we pointed out, are bundles of axons, all right, with the connective tissue that holds everything together and stabilizes it. Okay? An example here is the dorsal root ganglia. These are the nerve cell bodies of afferent nerves. Okay? These nerve fibers bring information into the central nervous system so they can move up to the brainstem and the brain. All right? There are other examples of ganglia that are in other locations in the body. Okay? Cranial nerves run from the brain or brainstem to structures in the head and neck, while spinal nerves are below the 12 cranial nerves. There's 31 pair, and they are all mixed nerves. Okay? And then you can see here the classification of sensory, motor, and mixed, and what they mean. Okay? And the definition of ganglion. Neurons are excitable, which means they respond to a stimulus, which can change the cell's membrane potential, and they have conductivity, which means they can propagate an electrical signal, so it can move from one location to another, and the speeds with which that happens will vary according to the types of nerve fibers. The speeds can be as slow as 5 miles an hour, they can be as fast as 250. All right, it all depends on the type of nerve we're talking about. They have conductivity, which means they can move the electrical signal quite some distance, and that's due to the presence of voltage-gated channels in regions of the nerve cells called the axons and the axon hillocks. Okay, those are the only places in a nerve cell that can create and propagate an action potential. They have secretory property. They can release neurotransmitter onto their target and change the membrane potential of the target and potentially create an action potential in the target or influence the target to some type of activity, okay, as a result of changing that membrane potential. They live quite a long time. Okay, some will live your entire lifetime. Some will live many decades. Okay, the older we get, the fewer nerve cells we have, and that's one of the reasons that the brain shrinks, that we lose coordination, and that we have a tougher time accessing and storing information as we get older. That's because simply the neurons die off. Okay, with very few exceptions, nerve cells do not divide. They are amitotic, and that's one of the reasons that damage to nerve cells is basically permanent. If you destroy the nerve cell, it will not reproduce, and only in peripheral nerves is there potential for repair. The cell body, also known as the soma, has all the cellular machinery in it. It has the nucleus, the endoplasmic reticulum, the mitochondria. Um, it is capable of generating graded potentials, which are going to fade with distance and be proportional to the strength of the stimulus. Okay, and that's due to the types of ion channels that are present on the soma and little extensions off the soma that are called dendrites. Okay, they contain Nissl bodies, which are made up of ribosomes, and they stain purple when treated to look at under a microscope slide. The dendrites are short, unmyelinated, tiny processes that come off the cell body that increase the surface area of the cell body so that it can receive chemical signals that generate graded potentials that could lead to an action potential. The axon is the long process that comes off the cell body. It begins at a region called the axon hillock, and it ends at the axon terminal, which terminate in what are called synaptic boutons, or terminal boutons. The terminal boutons contain vesicles that have neurotransmitter in them, which undergo exocytosis when an action potential arrives at the axon terminal. The release of the neurotransmitter causes it to spray onto its target, and that produces a change in the membrane potential on the target, which could result in an action potential in some cases, or a particular response in the target as a result of the change in membrane potential. The cytoplasm inside the axon is called axoplasm, and the membrane is called the axolemma. There are sometimes branches off the axons that are called collaterals. And the telodendria are it's just a collective term for the many axon terminals that branch off the end of the axon so that it can hit many targets. Okay. The cytoskeleton is made up of microfilaments, intermediate filaments, and microtubules. The intermediate filaments are called in the neuron neurofilaments, and they aggregate to form neurofibrils and provide strength to the cytoskeleton of the neuron. And you can see here an example of a typical neuron. The soma is located here, right? These short extensions are the dendrites. This long extension is the axon, okay? This would be a collateral. This white material is called the myelin sheath, okay? This happens to be a peripheral nerve neuron, okay? We can tell that because the myelin sheath here is made up of what are called Schwann cells that wrap around the axon in order to insulate and speed up the action potential so that it can get to its destination more quickly. It also protects the axon. And you can see the hillock up here where the action potentials are created. And here you can see the synapse, which is the tiny gap between the axon terminal and the target, okay? And this is where graded potentials are produced that potentially could generate an action potential. And this is just an actual slide of a stained neuron, okay? Looks kind of like a spider. Because the axon is so long, in a nerve cell, you can have a cell length of a foot or longer, which is pretty long for one cell. Anterograde transport is going from the cell body to the axon terminals, and the retrograde transport goes in the other direction, okay? This can occur both by fast or slow axonal transport, which we use different mechanism. Fast axonal transport is about 400 millimeters a day, and that is movement along the microtubules powered by ATP. Anterograde and retrograde motion can occur. Okay. Anterograde transport includes the movement of vesicles, organelles, and glycoprotein, and retrograde used vesicles and toxins and harmful material, okay, such as viruses, which can hitch a ride on retrograde transport. Slow axonal transport occurs at 1/100th or 1/1000th that speed, and results from the flow of axoplasm. This is called cytoplasmic streaming. Substances move only in an anterograde direction, and these include enzymes and cytoskeletal material and axoplasm. You can classify neurons by the number of extensions that come off of the soma. Okay, multipolar neurons have more than 2 extensions coming off the soma. Bipolars have only 2 coming off the soma, and unipolar have a single extension coming off the soma, which splits into a peripheral process that generates receptive dendrites and a central process that generates synaptic knobs that are located inside the central nervous system and are found in a region called the dorsal horn of the gray matter of the spinal cord. Anaxonic neurons have dendrites and do not have axons. Okay? Different types of neurons based on their function include sensory neurons, which conduct input from receptors to the central nervous system. Most are unipolar. Okay, these are located primarily in the dorsal root ganglia. Okay, motor neurons conduct output from the central nervous system to somatic and visceral targets, and all are multipolar. And interneurons are located entirely in the central nervous system, and what they do is process the electrical information before they send it on to its next location. They're primarily in material known as gray matter in the central nervous system, and are generally multipolar, and they are 99% of the neurons in the body. Okay, so you can see examples of most of these here. Okay, you've got unipolars here in the dorsal root ganglia. You got multipolars here in the motor efferents. Okay, and then you've got some more multipolars here in the gray matter, the dorsal horn of the spinal cord. The bipolars, where would you find them? You'd find them in the retina of the eye, okay, which is the part that takes light and turns it into electrical information that we eventually interpret as vision. And we also find that in the olfactory epithelia in the roof of the nasal cavity, and that is where we take chemicals that are dissolved in air and then use that to interpret odor, interpret smell. A synapse is just the gap between the axon terminal and the surface of the target. And this is where the neurotransmitter released by exocytosis drifts across a tiny gap and attaches to ligand-gated ion channels on the target in order to produce a change in membrane potential, which is called a graded potential. Important point here is that the synapse is the slowest part of any nerve pathway. So the more synapses a nerve pathway has, the slower that nerve pathway is going to be. An electrical synapse is a physical joining of adjacent cells. Gap junctions, which are tunnels that link the cells together, allow current to move quickly from one cell to the next. Examples of gap junctions include in smooth muscle and in cardiac muscle. Okay, there are also some examples of gap junctions in certain nerve cells. In the chemical synapse, the presynaptic neuron's axon terminal is going to generate the signal, while the postsynaptic neuron will receive it. The synaptic cleft is just the gap between the axon terminal and the surface of the target. The events of synaptic communication include the arrival of an action potential at the end of the axon, the movement of calcium through calcium channels into the end of the axon, which triggers exocytosis of vesicles that are filled with neurotransmitter, the release of neurotransmitter from the presynaptic membrane. The neurotransmitter then drifts across the synapse and then binds to ligand-gated ion channels on the surface of the target, and that opens up ion channels and causes ion movement, which could either be hyperpolarizing or depolarizing. Okay, if it's depolarizing, it makes it more likely that an action potential will be produced and an electrical signal will be sent down the pathway, and if it's hyperpolarizing, it makes it less likely. Okay? The glial cells you could think of as the personal assistants to the neurons, and basically they're capable of mitosis, and they do all the work that a nerve cell doesn't have time to do. They protect the nerve cell, they rescue nutrients from the blood and give it to the nerve cell, they remove waste products, they insulate the axons of the nerve cell to speed up electrical signals, they provide immune function for the nerve cell, okay, and they create something called the blood-brain barrier, which is a way to isolate the amitotic neurons from harmful material in the blood that is polar. Okay, and they also produce a liquid called cerebrospinal fluid that allows the central nervous system structures to float in material that creates the proper pH and ion concentration and temperature for the neurons to do their job. Okay, so this is a filtrate of blood that flows through hollow chambers inside the central nervous system, bathes the entire outside of the central nervous system, and then returns to the blood as quickly as it is produced. Okay, so it has a flushing action, and it also brings in ions and serves as a way for nonpolar material to diffuse from the blood supply to the nerve cells. Okay. In some cases, the glial cells can divide and form a mass called a benign tumor, and this is what you usually read about when you read that somebody has a brain tumor. And the danger of this, of course, is that the mass of the tumor can increase to the point where you crush critical central nervous structures against the bony outer covering and destroy them. Okay, so even though a benign tumor doesn't have the ability to spread through the bloodstream and set up secondary tumors in other locations, it is nonetheless a huge risk. Okay, the types of glial cells include astrocytes, which have processes that end in perivascular feet. These are the most abundant glial cells in the central nervous system, and they form the blood-brain barrier by wrapping their feet around brain capillaries, and this controls the substance's access to the brain. Okay, the ependymal cells also assist in this because they create the cerebrospinal fluid that we talked about earlier from the blood, but they also isolate material in the blood that would be harmful if it is polar from getting into the brain and causing damage. It regulates the fluid composition around the neurons, such as the potassium concentration, and it structurally supports the neurons. It aids in neuronal development, and it can take up space left by dying neurons. Okay? They get their name because they have a star shape to them. Ependymal cells line cavities in the brain and spinal cord and are part of a layer known as the choroid plexus, where the cerebrospinal fluid is produced in the roof of the ventricles of the brain. There are a total of 4 of those. Okay? The microglia are the only immune glial cells, and they protect against pathogens. They clean up dead cells and debris. Okay, but they're not nearly as effective at protecting against disease as the tissues outside the central nervous system. And this is one of the reasons we worry if a virus or a bacteria or some other pathogen gets in there, it can cause a lot of damage. Okay, the oligodendrocyte's job is to insulate and brace the axons of the neurons so we can speed up the electrical signal, provide a framework for the axons to hang on, okay, and we can insulate the axons. You can see examples of the different types of glial cells here. Okay, in the peripheral nervous system, satellite cells wrap around nerve cell bodies and nourish and protect them, while Schwann cells perform the same job in the peripheral nervous system that the oligodendrocytes perform in the central nervous system. Basically, they insulate and protect the axons and also speed up the electrical signal. And you can see examples of each of these here. Okay. When we have a tumor in the central nervous system, it's generally what's called a glioma. Okay, the glioma comes from a mutation in the glial cell that causes unregulated cell growth. The tissue mass begins to increase in size, and the result is crushing the nearby nerve cells. In some cases, they're malignant. Usually, though, that isn't the case. Right? But even if they're benign, they are going to produce damage to the surrounding tissue because of the increase in size. Okay, how do we deal with them? Radiation, chemo. Okay, in some cases we attempt surgery. All right, but it's a difficult task. Myelination is just the process of protecting the axons. And what happens here is that the axon initially is unmyelinated, and then in the peripheral nervous system, the neurolemmacyte wraps around the axon, and then the edge of the neurolemmacyte with the myelin in it progressively grows and advances and sort of wraps around the axon jelly roll style, and the nucleus of the Schwann cell and the cytoplasm get pushed to the outer periphery, okay, and that forms something known as the neurolemma. A neurolemmacyte can myelinate only a millimeter of an axon, so you need several in order to go the whole distance of the axon. Okay, so this is again in the peripheral nervous system because we're talking about Schwann cells here. The gaps between neurolemmacytes are nodes of Ravnier, and that's where new action potentials are produced after the initial action potential generated at the hillock is generated as a result of the effect of the graded potentials on that region. Okay, and it's the wide diameter, highly myelinated fibers that are capable of the most rapid transmission. So here you can see how the myelination takes place. Right, you can see how the neurolemmacyte wraps, and then the leading edge wraps around. So the youngest part, okay, of the myelin sheath is the part that's closest to the axon, and the oldest part is out here in the periphery. Okay, and so you can see the organization here. Right, any damage to this will result in a slowing of the electrical signal and potentially causing the electrical signal not to make it to the end of the axon and thus not reach its target. Okay, this happens in conditions such as ALS and multiple sclerosis. In the central nervous system, it's the oligodendrocytes that do the myelination. They can wrap around several different axons at the same time. It takes several oligodendrocytes to myelinate the entire axon, but a single oligodendrocyte will myelinate several axons at the same time. Okay, there's no neurolemma, and the nodes are the same as the nodes in the peripheral nervous system. They're there to accelerate the speed of the action potential from where it's created to where it gets to its target. Okay, there's another difference in that the myelin sheath of the central nervous system isn't capable of supporting regrowth of the axons if they're severed, whereas in the peripheral nervous system it can. Okay, so in the peripheral nervous system with unmyelinated axons, the axon sits in a depressed portion of the neurolemmacyte, right? Well, in the central nervous system, the unmyelinated axons are not associated with the oligodendrocytes at all. Okay, so you can see here in the unmyelinated axon, and then in the in the case where they wrap, right? You can see how they distribute, okay? The fact, again, that there's connective tissue in the nerves means that you're not going to have crosstalk between the electrical signals despite whether or not myelin is present, okay? So even in the event that there is little to no association of myelinated material with the axon, the integrity of the electrical signal won't be affected. We talked about multiple sclerosis. What happens here is that for reasons we don't understand, our immune system invades or breaches the blood-brain barrier and attacks the myelin sheath of the upper motor neurons, which bring the electrical information from a region of the brain called the cerebral cortex down to what are called the lower motor neurons, which are the part of the peripheral nerves that go out to the muscle targets. What happens here is that the myelin sheath gets torn up, and eventually the motor signal does not make it to its target, and the muscles, as a result, lose tone and lose strength and atrophy. In Guillain-Barré, we see loss of myelin from peripheral nerves due to inflammation. The muscles weaken. This begins in the distal limbs and advances to proximal muscles, but most of the function can be recovered with relatively mild medical intervention, okay? Whereas MS, there's not a lot we can do for it. We can slow the progression, but we can't stop the disease. After traumatic injury, peripheral nervous system axons can regenerate provided the neuron cell body is intact, the neurolemma remains, and the ends of the nerves, the distal and the proximal ends of the nerve, line up so they're flush, okay? Regeneration is more likely if the damage isn't terribly extensive and the distance between the damage and the target is short, right? So again, regeneration is possible in the peripheral nervous system. It doesn't always happen, but there's the potential for it to happen, all right? So what happens here is that the axon is traumatized. Let's say it gets cut. The area proximal to the cut will seal off and swell up. Distal to the cut, the axon will disappear, and this is a process called Wallerian degeneration. The neurolemma remains intact because, again, that's created by the Schwann cells here. And the neurolemma and endoneurium are going to form a regeneration tube, and then the axon regenerates because of nerve growth factors that the neurolemmacytes produce, and then we reinnervate the target. This does not happen in the central nervous system, and the reason, again, is that there isn't a neurolemma, okay? And so we don't generate the growth factors, in addition to the fact that usually when there's an injury in the central nervous system, we end up developing scar tissue that prevents the regrowth of the axon and the reinnervation of the target. So you can see the steps of regeneration here, okay? And then there's the deal with the central nervous system, right? Regrowth is obstructed by scars that come from astrocytes and the immune system. There's a large number of axons in the central nervous system, and the oligodendrocytes don't promote the regrowth of the axon, okay? All right. Let's talk a little bit about nerve tissue. Up to this point, we've kind of gone through the basics of the organization of nervous tissue. So this is a chance to sort of walk through the whole picture. All right, so stay tuned. Okay, we talked earlier about the fact that the nerve cell is excitable. Okay, it'll respond to a stimulus. Could be a physical stimulus, a chemical stimulus, a voltage stimulus, by generating current that will flow a certain distance. Now, in the dendrites and the soma, that current only flows a limited distance, and the name for that type of current is a graded potential. Okay, and the graded potential can either be excitatory, which means it's depolarizing, or inhibitory, which means it's hyperpolarizing, right? And just because you get a depolarizing graded potential doesn't mean you're going to create an action potential. You have to reach a value at the axon hillock called the threshold voltage in order for that to happen. And that occurs as a result of the combined effect of many graded potentials at the same time. Okay? And that comes from the neighboring neurons that surround the cell that you're observing. Okay? They're always sending chemical signals to that neuron and changing membrane potentials, and whether we generate an action potential or not is going to depend on the net effect there. Okay? Now, in the nerve cell membrane on the dendrites and the soma, what we have primarily are ligand-gated ion channels, and we have pumps, right? We have the sodium-potassium pump, and we have calcium pumps, okay? The sodium, potassium, and the calcium pumps are throughout the cell. The ligand-gated ion channels are found in the dendrites and on the soma, okay? So it's a particular composition of the membrane in the dendrites and the soma that promotes the potential to produce a graded potential, okay? The pumps need ATP in order to run, all right, to do their job properly. The channels that we were talking about include leak channels, which are always open and allow the movement of ions all the time, okay? The membrane is much more permeable to potassium than it is to sodium, all right? The chemically gated channels open when a ligand, a chemical, binds on the cytoplasmic side of the channel and pops open a door that lets ions flow through it, so it lets current move through a hole in the membrane. And voltage-gated channels pop when a particular voltage is achieved in that membrane and allows current to flow, okay? And then there are physically gated channels which respond to a particular physical force, okay, like pressure or tension, okay, or in some cases vibration. Voltage-gated sodium channels have three states, okay? The resting state, where both their activation and inactivation gates are closed and there's no flow of sodium through that channel. The activated state, where the activation gate is open and the inactivation gate is open and sodium flows through the channel, so there's a sodium current. And the inactivated state, where the inactivation gate is closed and the activation gate is open and no sodium flows through the gate in that case either, okay? So those are the three states of the voltage-gated sodium channels. Now you might ask, well, where do we find these on the neuron? Everywhere from the axon hillock to the axon terminal, okay? You don't really find them in the dendrites and the soma, all right? Other channels that are gated by different modalities are normally closed, but they open when their stimulus arrives, and they're found in the membranes of sensory neurons that respond to a particular stimulus. Examples, right? Some neurons in the skin have channels that respond to mechanical pressure, tension, light touch, all right? So we're all examples of mechanically gated channels. The entire plasma membrane of the neuron contains leak channels and sodium-potassium pumps, which are responsible for maintaining the resting membrane potential. Okay, but there's way more potassium than there are sodium channels. All right, and the sodium-potassium pump, as you remember, burns a single ATP and pumps out 3 sodiums to the outside of the cell for every 2 potassiums it pumps in. The net effect of this is that when there isn't anything else going on other than these pumps and these leak channels, that the inside of the nerve cell is negative compared to the outside. All right, and that is a form of potential energy, just like the charge in a battery, and that can be used to do work. Okay, and the work happens when current flows, when charged particles move from one place to another. Okay. The receptor segment of a nerve cell, which is the dendrite in the cell body, contains the ligand-gated channels. The axon hillock contains the voltage-gated sodium and potassium channels, as does the conductive segment, okay? And the transmissive segment, which is the secretory part, is going to also contain voltage-gated calcium channels. It's the movement of calcium into the synaptic terminals that results in the release of neurotransmitter by exocytosis. Okay? So you can see the different kind of channels and where they are located. Okay? So in a sense, all right, the nerve cell is a little like a quilt. All right? A quilt is a blanket made from different types of material from different sources that have different properties. And here, the membrane of the nerve cell is like a quilt. Right? The membrane on the soma and the dendrites has one particular composition, and the membrane from the hillock to the axon terminal has a different kind of composition. And so that's why you can only make an action potential in the axon, but you can make graded potentials in the dendrites and the soma. Okay? And then there's the calcium channels down there at the axon terminals. Okay? Some basic principles of electricity. Okay? Very important that we understand when we're talking about electricity. The equation is V equals IR, VIR. Okay? Where V is voltage, I is current, and R is resistance. Okay? All right. Now we can rearrange this, okay, to say that current is equal to voltage over resistance. It's the same equation. Okay? It's just been rearranged. Okay? And what that tells us is that there's more current flow when there's less resistance, and there's more current flow when there's a greater voltage difference. Okay? Important to keep that in mind. So low resistance means rapid flow of current, and high resistance means slow flow of current. Okay? Current is the movement of charged particles. Voltage is what we call the electron motive force. Okay, and it's measured in volts or millivolts, and it's basically the push that allows the flow to happen. Okay, but the flow isn't going to happen until two regions that differ in voltage are physically connected to each other. And then the resistance is just anything that opposes that movement. Okay, Ohm's law, we just stated it on the previous page, right? V equal IR, right? Or I equal V over R. All right. In neurons, charged particles are ions, and the current's generated when the ions move, and the voltage exists across the membrane due to the unequal distribution of ions, and that's created by several things. Okay, that's created by the difference in leakage between potassium and sodium across the membrane, and that's also created by the fact that the sodium-potassium pump pushes out more sodium than it pulls in potassium. And the effect of that is to create a negative charge inside the cell compared to outside the cell. And also another contributing factor is that the large negatively charged proteins are locked inside the cell and can't get out. Okay. The membrane has resistance to ion flow, and this resistance changes due to the actions of the gated channels. Resistance will decrease when the channels are open. Right? When the channels are open, ions will flow across the membrane from the outside to the inside. Okay? And when the channels are closed, they won't. All right? We should also point out, right, that not only can ions flow from the outside to the inside of the cell, but they can also flow across the inside of the cell. All right? So they can move from the soma towards the hillock, and then eventually from the hillock down the axon. Okay? So you got different kinds of movement going on here. Okay? At rest, the ions are unevenly distributed across the plasma membrane due to the action of the pumps. Higher concentration of potassium in the cytosol versus in the cytoplasm. Higher concentration of sodium outside the cell than inside the cell. Same with chloride, and same with calcium. Okay? All these are high outside and low inside. There's different reasons for this. Okay? The sodium's high outside because of the action of the pump and the difference in the permeability of the membrane to sodium. The chloride's high on the outside because the negative charge, follows the positive charge out of the cell. And the calcium's high on the outside because high concentrations of calcium inside the cell would form crystals that would be difficult to dissolve. Okay? So it's better to keep the calcium outside than inside. Okay? In addition to the fact that there's a difference in permeability. Okay? Gated channels are closed in the functional segments of the cell, and there's an electrical charge difference called the resting membrane potential, which is typically around negative 70 millivolts, which we can measure by putting electrodes inside the cell and outside the cell. And that represents the push that will move ions from one side of the membrane to the other. Right? The greater the difference in voltage, the more rapid the current flow is going to be. Okay, so you can see here at rest, quote unquote, all right, what the state of the neuron is. Okay, potassium diffusion is critical in setting the resting membrane potential. Potassium diffuses out of the cell because of the concentration gradient and the fact that the cell membrane is more permeable to potassium than it is to sodium. Okay, the diffusion is outward, but it's limited by the electrical gradient. Okay, so there's the charge interaction that will eventually limit the movement of the potassium out because opposite charges attract each other. Right, the inside of the cell is negative. Since there are relatively few sodium channels, sodium influences the resting membrane potential. Sodium likes to diffuse in because of its concentration gradient, but there's not nearly as many leak channels, so that means that the overall state at rest, and by at rest we don't mean chemical processes have stopped, we just means we're not generating a graded or an action potential, is negative 70 millivolts. Okay. The role of the pumps is to maintain what the leakage would eventually dissipate. Okay, so if we didn't have a pump, if we didn't have these pumps in there working, eventually the movement of sodium and potassium would mean that there would be no potential difference across the membrane, and the result would be that there's no electron motive force, which means that there's no potential energy stored in the membrane, which means we can't use it to do any work. Okay, and so that's the state of things at rest. Okay, so you can see the voltmeter here and the pump and all that. Okay, so let's take a look at the neuron at rest.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So what you're looking at here is just an overview of the anatomy of a neuron, showing you the soma, the dendrites, the hillock, the axon, and the telodendria and synaptic knobs at the end of the telodendria. This is the secretory portion, okay, in purple. This is where we release neurotransmitter. The action potential is generated from the hillock and moves down the axon to the axon terminals, and the graded potentials that create the action potential occur on the dendrites and the soma, okay, and it's their net effect on the hillock that determines whether or not we generate an action potential. In the receptive segment, we have to understand that because of the composition of the membrane, we have large numbers of either hyperpolarizing or depolarizing stimuli that result from binding to neurotransmitters from neighboring neurons, okay? So these are where axon terminals come in close contact to the dendrites or the soma, okay? And we talk about the fact that these graded potentials are short-lived. They are proportional to the size of the stimulus, and they can either be excitatory or inhibitory, okay? These postsynaptic potentials are going to have a net effect because they will create local currents that will flow towards the hillock, okay? And only if we reach threshold voltage at the hillock will we generate an action potential, okay? Now, one of the things we have to understand is that we can produce an effect that gets us to the threshold voltage at the hillock in a variety of different ways, okay? We can either have excitatory postsynaptic potentials that are the result of sodium or calcium entry into the cell, okay? Or we can have inhibitory potentials that are the result of chloride entry or potassium exit from the cell, okay? In an excitatory postsynaptic potential, that region of the membrane becomes less negative and more likely to produce an action potential, okay, at the hillock, not in the region where it's created, okay? So this is all the result of current flow, okay? In an inhibitory postsynaptic potential, we're less likely to generate an action potential. And it's the net effect of all of these that's going to determine whether or not we reach a threshold voltage at the hillock that lets us generate an action potential. So take a listen to this summary of what we call spatial and temporal summation.
Before any stimulation, the neuron is at rest. Recall that the resting membrane potential of a typical neuron is negative 70 millivolts. This potential depends on the distribution of sodium and potassium ions across the plasma membrane and their movement via leak channels and the sodium-potassium ion pumps. Stimuli are detected by the receptive region of the neuron, which generally consists of the dendrites and cell body. When a neuron is stimulated, its resting membrane potential changes because ions begin to cross the plasma membrane through gated ion channels. If ligand-gated sodium ion channels open, sodium ions follow their electrochemical gradient and enter the cytosol. As the inside of the neuron gains positive charges, it becomes less negative, a change called depolarization. If ligand-gated potassium ion channels open instead, potassium ions follow their electrochemical gradient out of the cell, and the cell loses positive charges. This causes the membrane potential to become more negative than it is at rest, a change called hyperpolarization. Hyperpolarization can also be caused by the movement of negatively charged ions, such as chloride, into the cell. Small, local changes in the membrane potential are called local potentials or graded potentials. They can be depolarizing and therefore cause positive charges to enter the cytosol and make the membrane potential less negative. Alternatively, they can be hyperpolarizing and make the membrane potential more negative. Local potentials also vary in strength, depending on the length of stimulation and the number of ion channels that open. We can compare local potentials to the ripples caused by a pebble thrown into a pond. A larger stimulus, or pebble, will cause a larger ripple effect, and a smaller stimulus, or pebble, will cause a smaller ripple effect. Another important feature of local potentials is that they are decremental, meaning that they weaken as they get farther away from the source of stimulation, and so are only capable of traveling over short distances. If we use our pebble analogy again, as ripples move away from the point where the pebble hit the water, they diminish in size and eventually disappear. For this reason, local potentials cannot be used for long-distance signaling. A single neuron can receive many incoming stimuli in the form of depolarizing and hyperpolarizing local potentials. These local potentials combine as they spread across the membrane of the neuron. The sum of these potentials determines whether or not an action potential is generated. We will discuss that effect in the action potential animation that follows. Recall that all action potentials that reach the axon terminal involve depolarization of the same size. How is it, then, that the effect on the postsynaptic cell can vary? First, the frequency with which action potentials arrive at the axon terminal determines how much neurotransmitter is released. Also, in the nervous system, multiple presynaptic neurons can stimulate one postsynaptic neuron, and their effect will be cumulative. Finally, depending on which neurotransmitter is released and the types of receptors that are present on the postsynaptic neuron, the effect can be inhibitory or excitatory. We will now focus on excitatory postsynaptic potentials, or EPSPs, and inhibitory postsynaptic potentials, or IPSPs. Postsynaptic potentials are local potentials in the postsynaptic neuron. If they are excitatory or depolarizing, they will bring the membrane potential of the postsynaptic neuron closer to threshold. This makes the neuron more likely to fire an action potential. EPSPs typically result from the entry of positively charged ions, such as sodium or calcium ions, via ligand-gated cation channels. If enough excitatory potentials occur in the postsynaptic neuron so that its axon hillock reaches threshold, an action potential will be generated. In contrast, inhibitory postsynaptic potentials, or IPSPs, cause a small hyperpolarization, so the membrane potential becomes more negative, moving it further away from threshold. IPSPs generally result from one of two events. 1. The loss of positively charged potassium ions due to the opening of ligand-gated potassium ion channels, or 2. The gain of negatively charged chloride ions due to the opening of ligand-gated chloride ion channels. IPSPs make the neuron less likely to reach threshold and therefore less likely to fire an action potential. Recall from our discussion of local potentials that a single neuron can receive many incoming stimuli in the form of depolarizing and hyperpolarizing local potentials. If these local potentials are caused by synaptic stimulation, they are called postsynaptic potentials. The neuron then integrates all incoming stimuli And if threshold is reached, an action potential will be fired to propagate the signal farther down that neural pathway. If threshold is not reached, then the signal will stop at that neuron.
So as you can see, it's the summation of all of these different potentials, either as a result of spatial or temporal effects, or a combination of both, that will potentially produce threshold voltage at the hillock. And if that threshold voltage is produced, then an action potential will be generated. All right. Now, if we remain at the threshold voltage long enough, all right, we can make more than one action potential. Okay, and that would come from a more profound effect from these graded potentials on the hillock, maintaining that threshold voltage for a longer period of time. Okay, the result of generating more action potentials is that this is interpreted by the target as a stronger electrical signal. Okay, whether it be, say, a muscle contracting more powerfully, or in the brain, a sensation being more intense. Okay, so the more action potentials you have per unit of time, the more powerful the command is or the stimulus is. Okay, so those are the two types of summation. Okay, spatial and temporal, as we talked about earlier. All right, and you can see again how the net effects work. Okay, just as indicated in the previous video, in spatial summation, you can have multiple effects at the same time, okay, generating some currents that are excitatory or some that are inhibitory, whereas in temporal summation, we can hit the same area multiple times and generate a stronger net effect at the hillock, okay, and we can, of course, combine those two, okay. In the initial segment, which is the hillock, if we reach threshold voltage, we will generate an action potential, and it will propagate to the end of the axon and result in the release of neurotransmitter. Okay? If we do not reach threshold, then we won't manage to do that. Okay? So it's all up to whether or not we're capable of reaching that threshold voltage as a result of the excitatory potentials outweighing the inhibitory potentials, and having enough of those excitatory potentials to reach threshold voltage. Okay, so again, this is all about current flow. Okay, and we have to have enough current flow across the inside of the membrane to get the hillock to threshold. Okay, the action potential is a unique event. It can only occur in the nerve cell between the hillock and the axon terminal. Okay, it begins with depolarization of a segment of membrane as a result of sodium entry. Then what will happen is sodium gates will shut, potassium gates will open, repolarization will occur, and even hyperpolarization. And then that segment of membrane will reset to its original distribution of ions across the membrane as the leak channels and the pumps do their work. Okay? The action potential always goes in one direction, one magnitude, and one speed from where it's produced to where it stops. And as a result of that, it is a long distance electrical signal. Okay? So you can see the changes that take place, right? At resting potential, the gated channels are shut. As we get towards threshold, voltage gated channels pop, sodium comes in, depolarization occurs, okay, and then we reach plus 30, we shut the sodium gates, we open the potassium gates, the potassium will rush out, repolarization will take place, and even hyperpolarization, okay, and then we will return back to the resting membrane potential. So you can see those events here, right? Here are the graded potentials that are coming from the dendrites and the soma affecting the hillock, right? So here we go, here we go, here we go, and once we get to threshold, bang, we open the sodium channels, we rapidly depolarize, then we close the sodiums, open the potassium rushes out, we even hyperpolarize a bit, and then the leak channels and the pumps reset everything back to its original voltage, okay? And you can see that right there, okay? Now, an important point is that there is a period of time which is called the absolute refractory period that runs from where this dotted line crosses the red curve to where this dotted line crosses the red curve, where it's not possible to make another action potential until things reset, okay? So what happens during the absolute refractory period is that we force the action potential to go in one direction, okay? The result is that we move it from the hillock to the terminal because it can't back up and affect the previous segment of membrane because it's not capable of producing another action potential at that point. Now there is a period of time after the absolute refractory period, which is called the relative refractory period. You can see that here, where a significantly strong stimulus could produce another action potential, okay? So that's the refractory period, right? And it's defined by the time between when the activation gates of sodium open to when the activation gates of sodium have closed again. And that happens at threshold voltage. The relative refractory period is the hyperpolarizing period afterwards, okay? So you can see again the periods broken down for absolute and relative refractory period, okay? The bottom line is that this is going to define the number of action potentials per second that the axon can create, okay? The closest you can hug those action potentials together is the length of the absolute refractory period. So a significantly strong stimulus can generate multiple action potentials that are basically the length of time from the depolarizing to the repolarizing wave where it intersects the threshold voltage, okay? Now, that said, all right, let's take a look at the generation of an action potential, all right, and the events that follow.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Now, in the conductive segment of the neuron, which is from the hillock down to the axon terminal, we have the movement of the action potential from its source to its destination. There's two ways that this can happen. In continuous conduction, every square inch of the axon is involved in the creation and the movement of the action potential from one membrane segment to the next. Okay? Continuous conduction speeds can run anywhere from 5 miles an hour to maybe 15. Okay? So on the scale of things, they're relatively slow electrical signals. All right? Whereas in saltatory conduction, what happens is that segments of the axon are insulated and as a result have very little resistance to current flow, which means that the current generated in the unmyelinated segments of the axon moves very quickly under the myelinated segments and generates a new action potential at the next gap in the myelin sheath, which is called a node of Ravnier. As a result, the myelination speeds up the action potential by an order of magnitude. So top speeds for myelinated wide diameter axons are around 250 miles an hour. And we see that in somatic motor neurons and in somatosensory neurons, okay, where the speed is critical for the information to make it to its destination and still be effective, okay? So the idea with the saltatory conduction is that it's faster, okay? The myelinated cells use less ATP to maintain resting potential, all right? And it does require additional resources, okay? Because of the speed with which the signal is moving, it's possible to move a lot more signals down a myelinated axon than down an unmyelinated one, okay? What this means is that the body is putting the high-speed cabling to its most effective use, all right? We don't need 250 mile an hour nerve fibers for everything that goes on in the body, all right? That would be a waste of resources. So we save the high-speed lines for the stuff that needs it, and the slower speeds are for things like autonomic efferent neurons, okay? Some special sensory neurons, and so on, okay? You can see here in the propagation of an action potential how this is speeded up versus continuous, right? And it's all about resistance, right? The resistance in the myelinated axon is low in the areas that are insulated. And so the speed of the current is way faster here, okay? Whereas in continuous conduction, the speed is slower due to the fact that there's resistance from like charges on the other side of the axon, or because of the periphery of the axon, that will reduce the speed with which the current flows, in addition to the fact that the axon itself is a little bit leaky, okay? There's no leakage in these insulated regions, okay? So slow speeds here, high speeds here, okay?
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
The transmissive segment at the synaptic knob, when the action potential reaches it, what will happen is the calcium will flow in, exocytosis of neurotransmitter-filled vesicles will follow, neurotransmitter will be released and will bind the ligand-gated ion channels on the target membrane. Okay, so basically we spray neurotransmitter on the target, and the result is that we create a change in membrane potential that has the potential to lead to an action potential. And that's what goes on at the chemical synapse, which is the tiny gap between the axon terminal and the target cell membrane, as you can see here. Okay, but it's the influx of calcium that's critical to the release of neurotransmitter in those vesicles. Okay, so when we compare graded versus action potentials, the thing that we notice about a graded potential is it can be either excitatory or inhibitory. It only happens on the dendrites and the soma. Okay, the length of the current flow is limited because it's proportional to the strength of the stimulus, and it fades with distance. Okay, while action potentials move at one speed, one direction, and one magnitude from where they're created to where they stop. All right, and that happens only between the hillock and the axon terminal. So remember, right, that it's the difference in the composition of the membrane in those regions of the neuron that makes it so that action potentials only occur between the hillock and the terminal, while graded potentials occur on the dendrites and the soma. Okay? It's not surprising that the speed of an action potential depends on the axon's diameter and the degree of myelination that it has. Thicker myelinated fibers are the fastest, while narrow unmyelinated fibers are the slowest. Okay? Group A fibers are the 250 mile an hour guys, okay? And those are somatosensory neurons and somatic motors, okay? Group B, about 25 miles an hour, okay? And then Group C, around between 2 and 5 miles an hour, okay? So again, the uses for the slower fibers: visceral neurons, somatosensory neurons from the skin, okay? Autonomic neurons would be included in visceral neurons. And the idea here is that you don't need those high speeds for those functions, okay? That would be a waste of resources. The frequency of the action potential determines the strength of the command or the strength of the stimulus as interpreted by the central nervous system. The more rapidly you generate the action potentials, the stronger they're perceived as a stimulus by the brain or the brain stem, right? Working with the hypothalamus. Or the stronger the command is when aimed at the target, okay? So if I hit muscle fibers with rapid volleys of action potentials that result in the release of neurotransmitter, that's going to create rapid volleys of action potential on the muscle fiber, and that's going to result in a stronger contraction, okay? Whereas if the frequency of the action potentials is way less, then the contraction won't be as powerful, okay? For neurons that can use different neurotransmitters, the frequency can influence the type of neurotransmitter that's used, okay? So we need to keep that in mind as well, okay? We can classify neurotransmitters based on their effect and their chemical composition. Neurotransmitters are synthesized by the neurons and stored in secretory vesicles in the synaptic terminals, and they're released when the calcium influx causes exocytosis. They then move across the synapse and bind to the ligand-gated ion channels on the target and produce a response, okay? There's about 100 different neurotransmitter types that have been classed into groups such as acetylcholine, which is made up of choline in an acetyl group, okay? It's used, for instance, in skeletal muscle contraction, all right? Biogenic amines, such as dopamine and serotonin, are made from amino acids. Tyrosine derivatives become catecholamines, and indoleamines are produced from histidine or tryptophan, all right? Amino acids can be converted into other types of neurotransmitters, such as glutamate and glycine and gamma-aminobutyric acid, or GABA, while neuropeptides are short proteins that include endorphins and substance P, which is responsible for...
Synaptic transmission is a transient event, like speaking a single word. As you've seen, it begins with an action potential arriving at the presynaptic terminal. Stimulation of the postsynaptic neuron will last as long as neurotransmitters are available to bind to postsynaptic receptors. When the neurotransmitter involved is no longer available, stimulation is terminated and the postsynaptic neuron returns to its resting state, allowing it to respond to future stimulation. We will now examine the three mechanisms that allow for termination of synaptic transmission. The first mechanism allows some neurotransmitters to simply diffuse away from the synaptic cleft. They might eventually diffuse into surrounding neuroglial cells. Another mechanism for synaptic transmission termination is degradation of the neurotransmitter by an enzyme in the synaptic cleft. An example is the enzyme acetylcholinesterase, which breaks down acetylcholine. The components of the degraded neurotransmitter are often recycled back to the presynaptic neuron, and then reused to synthesize more neurotransmitters. And finally, other neurotransmitters, such as dopamine and serotonin, are removed from the cleft by reuptake into the presynaptic terminal, a process facilitated by specific membrane transport proteins. Once inside the axon terminal, the neurotransmitters can be degraded by an enzyme or repackaged into vesicles and reused. Regardless of how the neurotransmitter is removed, once it is no longer in the synaptic cleft, synaptic transmission is terminated.
Propagation of the pain signal, okay? And they can be classed by their effect on their target. Excitatory neurotransmitters cause EPSPs and inhibitories cause IPSPs. Some neurotransmitters can excite certain targets and inhibit others. Now, why is that? Because of the type of ion channel they open, right? So, an example, you might have a particular neurotransmitter that opens a sodium channel on one target and generates an EPSP, and it might open a chloride channel on another target and cause an IPSP. Okay. Direct neurotransmitters bind the receptors, the ligand-gated channels, and pop them open, while indirect transmitters involve a G protein cascade that generates second messengers that will eventually open ion channels and produce responses in the target. Okay, so you can see the chemical structure classes and the functional classes here. Okay, note that acetylcholine is the neurotransmitter of choice in innervating skeletal muscle. The action potential triggers its release into the neuromuscular junction. The acetylcholine attaches to the postsynaptic receptor and is cleared by the action of an enzyme called acetylcholinesterase that breaks apart the acetyl group in the choline, and the choline and the acetate are taken up by the axon terminal and recycled to make new neurotransmitter. The effect on the target cell depends on the type of receptor. Nicotinic versus muscarinic. Okay, nicotinic acetylcholine receptors are generally excitatory, and muscarinic can be inhibitory. Okay. Other ways that we can clear neurotransmitter from the synaptic cleft, aside from enzyme breakdown, is that reuptake may occur. There are transporters that import the neurotransmitter. Some neurotransmitter will diffuse away and get reabsorbed by glia. The way that most psychoactive drugs exert their effects is by either increasing or decreasing the amount of time that the neurotransmitter spends in the synapse bound to the ligand-gated channel. Okay, an example is a group of drugs called SSRIs, or selective serotonin reuptake inhibitors. And what they do is they slow down the ability of the serotonin neurotransmitter to be soaked back up by the axon terminal that released it, and as a result, the effect of the serotonin lasts for a longer period of time. SSRIs are often used to relieve conditions such as anxiety and depression. Okay, so you can see here the action of acetylcholine on its target, right? The choline and the acetate are the breakdown products, and then they get reabsorbed and recycled. And that means that we use less time to resynthesize neurotransmitter, since you never know how quickly a new action potential will arrive. Okay? Neuromodulators alter the response of the neuron. Facilitators cause greater response in the postsynaptic neuron and may increase the amount of neurotransmitter in the cleft or the number of postsynaptic receptors, while inhibition will do the reverse. Okay? So this is sort of a set the table kind of chemical release. Other examples of neuromodulators: nitric oxide can be a neurotransmitter or a modulator. It is a gas. It is nonpolar, so it can cross the membrane without a transport protein or a special tunnel or gate. It's made and released by postsynaptic neurons in the brain, where it strengthens memory by causing presynaptic cells to become more prone to firing under the conditions that make that nerve pathway activate. The effects in the peripheral nervous system include blood vessel dilation in the reproductive organs. Cannabinoids influence the same receptors that marijuana does. These are nonpolar molecules that are released by postsynaptics, and they have effects on presynaptic neurotransmitter release, influencing the ability to store memory and enhancing appetite. Okay? The circuitry that goes into moving the electrical signals around in the central and peripheral nervous system can fall under 4 different types of groups. You can have converging and diverging circuits, you can have reverberating circuits, and you can have what are called parallel after discharge circuits. The pool can be localized or distributed in different regions of the central nervous system. In a converging circuit, many neurons impinge on a single target, which then has a single output. Where this might be useful are sensory inputs, neurons in the salivary nucleus that promote the preparation of the oral cavity for food, right? Sight, sounds, and smells of cooking, for instance, cause salivation. In a diverging circuit, we have the opposite situation. We have a single input that forks out to multiple targets. Examples include neurons in the brain that control stereotypical movements, okay, such as the muscles involved for walking, posture, and balance. Okay, so it's sort of like hitting an electrical program that executes itself, such as when you reset a printer by pushing a combination of buttons. Okay. In a reverberating circuit, what happens is that the output feeds back to neurons that are earlier in the pathway and causes that pathway to execute again, over and over again. All right. It's thought that this type of circuitry is responsible for the rhythmic innervation of the diaphragm that allows us to breathe while we're asleep. Okay. Well, parallel after discharge circuits involve inputs that move at the same time along several paths to a postsynaptic. The paths vary in the number of synapses and arrive at different times, and this is thought to be involved in higher cognition. So a lot of this is going on, for instance, in the frontal lobe of the brain. Okay? So I will see everybody in the next podcast. Please let me know if you have any questions, and I will contact you. I will see you in the next chapter. Thank you.